So I think I'm going to give you this talk based on the my among the different hats as biomedical engineering. I'm not sure this is going to work. Just the next slide, please. I don't have any disclosure. Next. So this is the definition. The nanotechnology is a science, engineering, and technology conducted at the nanoscale, which is about 1 to 100 nanometers. This is actually defined by the National Nanotechnology Initiative. So next. So if you look at this right side of the panel, this is a baseball about 10 to the 8th nanometer. You look at the glucose molecule about 1 nanometer, DNA 10, virus about 100 nanometers. The cancer cell is around here, which is about 15 to 50 micron. So if one cancer cell, it can contain hundreds of thousands of these nanoparticles in it. So basically these are all the nano materials has been working, liposome, dendrimer, gold nanoparticles. I'm going to talk about this one today is a little bit more, and nanorod, quantum dots. This is also more imaging agents, and then fluorine and the carbon nanotube and others. Next. So basically this nanotechnology can apply many areas, basically not necessarily cancer, but also medicine as a whole. Drug screening, drug delivery, detection, gene delivery, monitoring of the therapy, and many other things. Next. So I think there's already available drug in terms of the solubility issues, like Abraxane has been using that. But I think a second point is I'm going to focus today, which is non-specific delivery and targets. So many of these drugs, as you know, it can kill this cancer cell, of course, and also kill normal cells and makes a tremendous toxicity. In fact, this fact addressed by the nanotechnology. Many other things here, but I'm going to focus this. Next. So this is a concept of active and passive targeting. So passive targeting basically through the diffusion based on the concentration of the drug, but active basically you have formulated specific nanoparticles with a ligand, and then this ligand can bind to the receptors of the cell surface. So either these are blood vessels or tumor cells, and this is usually required energy. So this is a very important concept to how we can specifically target the cancer cells. Next. So this one actually our review papers a number of years ago, we published that. And basically if you have cancer cell have some receptors here, multiple receptor versus very small or none, then whatever nanoparticle formulated, which is conjugated with a ligand, can bind to the receptors. And these nanoparticles mainly go to the cancer cell, not much normal cell. This is a hypothesis kind of driven here, but I think we are more and more proven such a particles now available. So next. So this actually we reviewed very extensively over the next, last years about this concept. I think it is going to be really truly implicated in the cancer therapy next 5 to 10 years. Next. So here this is kind of partial list of the ligands we can formulate it with a nanoparticle. But here I just want to talk about the transferrin, which is a receptor, is a transferrin receptor, folic acid, and the folic acid receptor. And this is EGFR or EGF as a ligand and bind to the receptors. So these are three things I'm going to talk about today. Next. So this one basically what I just illustrated here, we wrote this paper a number of years ago about this tumor progression from normal to all the way to the invasive cancer metastasis through the pre-malignant change. Of course, a lot of genetic change. It is just a partial list here. But in terms of nanotechnology, actually not only therapeutics and even prevention. Recently, we published a paper using some of our natural compound formulated nanoparticle, and we just tested. It works very well. So I think nanotechnology can be applied all throughout this process. Next. So basically, this one actually siRNA delivery I'm going to talk to you today. you know that siRNA is a kind of a target gene silencing, and then eventually the protein synthesis decrease, and then cancer cell cannot survive. But problem is delivery. Actually, this siRNA is very sensitive and also easily degraded in the blood stream like that. So there's several ways we can deliver. One is the liposome-based delivery, polymer-based delivery, peptide or aptamer-based delivery. A lot of actually we're currently working in the nano field. Next. So one of the enzyme we are working on, this is a so-called the ribonucleotide reductase, which is a very important enzyme, the DNA synthesis here. There's two different RM1 and RM2 here. So we actually targeting this RM2 using this siRNA with actually effectively this is blocking this process. But delivery is still big issues here because this is easily degraded and it's not working in vivo or even in patients. Next. So basically, Dr. Mark Davis from Caltech, actually they formulated very interesting nanoparticle for delivery of siRNA targeting RM2. So this is a cyclodextrin as a backbone nanoparticle, and also pegylated, and then adamantine attached there, and then transferrin actually attached again. So this is look like this. Next. So basically this one published in Nature 2010, and they actually did a phase 1 trial among the after multiple in vivo studies like that. Then this is actually biopsy from the actual patient. This is a case of the melanoma case, and then biopsy and then actually stain with immunofluorescence. These nanoparticles well visualized. This is after one delivery, not much here, but you have a couple of signals here, and then another one, and also you have twice delivery. There's a lot of signals, you can see it. Next. So what they did is using the biopsy samples, actually they did sequence, and actually that fragment is about 200 base pair, and then when they sequence, exactly same site. This is a culture set as a control, and this is one sample from the patient. Actually, this is cleavage site exactly identical. So they can confirm after biopsy what they deliver and work in the tissue. Next. So with collaboration with the Mark Davis group, actually we look at the first RM2N transferrin receptor expression of these cell lines. So all head and neck cell lines, as you see here, quite well expressed in lung cancer cells, some of them too. Some of them are high, some of them are low. And also RM2 expression is all head and neck cell lines, and it's a very high expression. And then some of them lung cancer as well, but some minor expression here too. Next. So you look at the siRNA function, it works, and then this is cell growth, nicely inhibits cell growth. This is two head and neck cancer cell line to the lung cancer cell line, and then apoptosis assays increase nicely. Next. So also this is Western blot showing that RM2 nicely decreased, pop cleavage induced, P73, which is a P3 partner, nicely induced, AKT phosphorylation decrease, and this is the lung and head and neck cell lines. Next. So basically, based on this, we did in vivo experiment using the xenograft model, and here we have 3 different controls. Water control, unformulated siRNA, which is, as you see, that all degraded during the delivery to the target. And this is a control formulated, which doesn't work either. So this is 3 controls, and then this is a siRNA nanoparticles. It says 5 milligram per kg, 10 milligram per kg, nicely decrease the tumor growth here. This is all control, and blue, this one is the 5 milligram per kg. And the magenta color, this is 10 milligram per kg. Nicely inhibits tumor growth. This is at the time of experiment, showing the tumor size comparison. Next. So basically, 2007, we established head and neck cancer spore program at Emory, and we actually, one of the projects is targeting cell surface receptor with surface receptor targeting the nanotherapeutics. Next. So one of the things we look at is a folic acid receptor, which is also quite frequently expressed in the head and neck cancer. Here we look at about more than 100 samples of the patients, and then actually the folic acid expression is quite high, at least 50% or higher, and then some of them extremely high. So this is an excellent target. However, bone marrow, we didn't see. We look at the 10, but nobody actually expressed. In fact, this bone marrow, if you look at the folic acid receptor, some of them expression, but all folded down. So therefore, in vivo situation, they just don't express that. That is actually good for the, in fact, in the future in the patients. Next. So this is nanoparticle formulated. This is a heparin-based nanoparticle, folate, and then Taxol. So this is a final product like this. This is takes about almost 1 year to actually produce this nanoparticle. And then this drug, Taxol, actually inside, and then outside is actually hydrophilic and conjugate with the folic acid. Size is very excellent. If you have too small, less than 10 nanometer example, it's easily leaked through the kidney. And if you're too high, that's also problematic. So about the 56 nanometers is an ideal size. Next. So this is a colony formation assay. We'll look at it. This is a control cells. These are KB301 cells, which is a very high expression folic acid receptor on cell surface. The Taxol treated. This is a non-target nanoparticle. It's target nanoparticle. Clearly, you see the big difference in terms of cell growth inhibition. Next. And this is the animal model we tested. And just looking at the free Taxol versus this target nanoparticle, clearly you can see the big difference in cell tumor growth in these animals. And then actually we did the 3 of them and didn't show any single viable cells there. So this is a big difference in terms of cell growth inhibition here. Next. So next one, we are going to talk about something gold nanoparticle, which is actually another nice delivery. This is we published in 2007 with my group and Shuming Li's group from biomedical engineering. Next. So this nanoparticle about 50 or 60 nanometer, and this gold is actually conjugated with a Raman reporter first, and then pegylated later. And then we can conjugate it later with the different nanoparticles. Next, actually we use the EGFR single chain fragment right here. This one is just using as a ligand attached to the drug and a nanocarrier here. Next. Now this is the final kind of cartoon here showing this nanoparticle EGFR single chain sticking out and then supposedly bind to the EGFR positive cells alone. So in fact, it is true. We're using the TO686 high EGFR expressed cells, and then you see the very nice signal when you do the appropriate laser rate. And then H520, which is negative EGFR receptor there, and flat, we didn't see any. And also without ligand, of course, we don't see that. And then this is a control in the water tag. Next. So also we did an animal study, and basically this is a tumor injection to the flank, and then this is a laser hit it, and then we see the nice signals here. And the baseline, this is the liver, just a little background, and non-target one, didn't see anything there. Next. So basically now we are using this one, different nanosystem 2, iron oxide. This is not gold, and this is a single EGFR conjugated. And actually, we are currently working on conjugate with the drugs and also imaging agents together. So we can see that imaging and drug is kind of monitoring and then treat the cells. Next. So this is just showing that positive EGFR cells injected here in growing, and then we injected this, tag the EGFR particles and can nicely visualize. And this is a kind of negative EGFR. Both flank were injected, and of course the EGFR positive cells are only visualized by this technique. Next. So basically now NCI, Nanotechnology Alliance Network, has been, this is a second term now, 5 years. So about 10 years almost we did it. And basically now we have multiple institutions from East Coast, Midwest, Southwest, and West Coast. We have about more than 30 institutions participated. This is the NCI and Nanotechnology Alliance. And we have one project going on here, photothermal treatment of cancer with golden nanoparticle. This case is nanorod. And with the collaboration with Georgia Tech, Dr. Elside group, and my group. Next. And basically now we have 3 different golden nanoparticles. It's golden nanoparticle actually a lot of advantage. Because of the, if you look at the gold nanoparticles, very intense resonance scattering of the light. Therefore, we can see very well visualized. And also, if you look at the therapeutic effect, very high conversion rate of the energy to the heat. Therefore, if you hit even small energy, you can see the maximize and then killing the cells. So this is a nanorod, shell, and cage, but this is what we are working on right now. Next. So this is basically looking at the different ligand attached LGD, ATF, and also single chain EGFR. And you can see that those little bit yellowish and reddish color, that's the actual signals, and then the other is no signal there. But actually, this is also signal actually based on this ligand attached to the nanoparticles. Next. So they also will look at the long-term toxicity. A lot of people concerned about whether golden nanoparticles are really making any issues. So this is all the up to the 15 month, and you have all these organs. We don't have any damage there. And then EM study, also no damage up to the 15 months, which is translated human age of about 60 or 70 years old. Next. So this final, this one is toxicity, again, no. And then kidney, TBU, and creatinine, no toxicity up to the 15 months, no. Next. And this is therapeutic. If you have a control and then treat with golden nanorod, and nicely, this is even no drugs there, just a hit with the laser. We can see the very nicely tumor growth inhibition we can induce. Next. So ephemery, golden nanorod is no toxicity observed in the long term up to the 15 months. And also photothermal therapy, I think intra-tumor injection, I didn't give you much detailed data here versus the in vivo and the intravenous, but intra-tumor injection is much better. And also large animal studies now is planned, and the conjugation of anti-cancer drug onto this golden nanorod is next step. Next. So I think this is just a list of some of them clinical trials ongoing. These are earlier versions, kind of, this is not really first generation one, is approved that, but a lot of them still preclinical studies ongoing here. Next. So in conclusion, nanotechnology-driven drug discovery and delivery is a rapidly evolving science in cancer therapy and diagnostics. Nanotechnology can address many limitations of current drug discovery and delivery, and multi-component nanotherapeutics actually still some issues to go into the eventually approval by the regulatory agency, so we have to work on that. Nevertheless, nanotechnology-driven drug discovery and delivery is highly promising in future cancer therapy. Next. And nanoscience is a team science. We need a lot of different specialties work together in this area. Next. So I would say that incredibly small size can make a huge impact on future oncology and medicine. Next. This is my acknowledge and many folks in my laboratory and also Shuming Lee's lab, Georgia Tech's Mustafa Elside group, Mark Davis from Caltech, Bill Hunt from Georgia Tech, and many grant support. Thank you very much.